Hi everyone! From previous chapters, we have discussed how life has come to be according to the evolution and natural selection. We have also discussed some of the mechanisms as to how natural selection works and how to test whether a population is evolving through the Hardy-Weinberg equation. So now, for chapter 24, we will discuss how different species have come about through a process called speciation. Don't worry, it will be as long, not as long as the last chapter, because last chapter was like more than 2 hours long. But this one will be shorter. I promise that. All right, let's get started. In chapter 22, we talked about how Darwin became a naturalist and went on a journey around the world on the ship HMS Beagle, and that he has observed and collected many samples of plants and animals, especially from the Galapagos Islands, where he found species that existed nowhere else around the world. It was through observing closely at these species that he came up with the theory of natural selection and evolution. He then published a book on his discoveries called The Origin of Species. We now understand how his theories have come about from the previous chapters. So the evidence, and what was his evidence for natural selection, and also modern day evidence for natural selection, and also we learned how selection happened. So the next question to ask is, how do unique species have come about? For example, on the Galapagos Islands, there exists this flightless cormorant that is found nowhere else in the world except on the Galapagos Islands, right? So, how did this bird arrive at the islands if it cannot fly? That's why it's called the flightless cormorant, because it cannot fly. And you can see that its wings is very, very tiny, such that it cannot fly, or it doesn't have enough feathers or ergodynamics that will lift the bird up into the air. So Darwin thought it must have originated on the islands from ancestors that travel across from South America, because or else there would have never been this flightless bird or cormorant on the Galapagos Islands. Or so in another word, this flightless cormorant was not on the islands to begin with, but some of his ancestors that lived in South America decided to fly to the islands and start a new population there. And with time, it could have lost its ability to fly because it was not a trait that was suited for its survival in this particular environment, as the common theme we have discussed throughout these chapters. So today, we understand unique species have actually come about by the process called speciation. And speciation is a process by which one species is able to split into two species. This process repeatedly yielded new species that differ from the existing ones, and then this is what it essentially led to tremendous diversity of life. It also has helped explain why organisms of different species share features, which is what we talked about the key observation of life, the unity of life, meaning that everything descended from a common ancestor. So the diversity of life and unity of life is what we see here again about the, observation of life. Now, biologists compare morphology, which is the fancy word for appearance, physiology, biochemistry, and DNA sequences of different groups of organisms to divide them into discrete units that we now call species. The process of speciation is actually described by multiple concepts, because it's a very complex concept, and it's something very abstract as well, because we don't see a lot of that happening nowadays with macroevolution that I mentioned from the last chapter. We see a lot of microevolution, but not so much macroevolution. So the primary one that we'll discuss in this chapter is called biological species concept. We will mainly just focus on this one. This concept says that a species is a group of individuals in a population who has the potential to interbreed. Okay, so that is the key word there. They have the potential to interbreed in nature and produce viable, fertile offspring. But, but, okay, they cannot do so with other populations. This is sort of the um, main thing with the biological species concept is that these are the two key features, that we use. Remember that for this concept, species have the ability to interbreed and reproduce within the population. That is what it says. This is what defines as species according to the biological species concept. So hence, members of a biological species are reproductively compatible, and that's why they can interbreed. So for example, the eastern meadowlark and western meadowlark have similar body shapes and colors. You would almost think they look like the exact same bird, but they are distinct biological species because their songs and behaviors are so different that they don't even interbreed. You can see here in this picture that these two birds have yellow coloration, mainly on the ventral side of the body or the belly side of the body. And then there's some brown coloration on the dorsal side or the wings portion. They also have a black collared area on their chests from both birds. And because these two population of birds cannot interbreed, even though, even we're talking about, they says that they live in eastern and western parts of the world, but even if, let's say, if they were to meet in the wild, they would not interbreed. And that's why we actually consider these two birds as two different species. Everything counts, especially with the biological species concept, because they have to interbreed to produce fertile offspring in order for us to consider it as a species, the same species. So what keeps a population as the same species is the ongoing exchange of alleles. Exchange of alleles between individuals of a species. This is what we call in the previous chapter gene flow. So gene flow is what holds the gene pool of a species together. Therefore, those individuals resemble each other more than those of other species. And it is interbreeding that allows for gene flow to happen. According to the biological species concept, species are defined in terms of reproductive compatibility. Like we talked about with interbreeding, they have to be compatible in order to interbreed. And hence, formation of new species will require what we call reproductive isolation, which means that there exists some sort of barrier that prevents reproduction between two species, kind of like what we talked about with the birds earlier in the previous slide, that their songs and behavior are different and hence they would never interbreed. That's a form of reproductive isolation. So for example, there can be biological barriers that impede members of two species from interbreeding and producing fertile offspring. Because that's a common theme here. They have to have the ability to interbreed and produce those fertile offspring. And such barriers block gene flow between species and limit formation of hybrids. Well, what do you mean by hybrids? Hybrids are offspring that come from mating of two different species. Okay? So which means these barriers have to exist to block the gene flow, and there is limitation of formation of hybrids. That's what we mean by reproductive isolation. Now, these barriers can be classified as two separate categories. One being pre-zygotic barriers, meaning that we're blocking or there is some sort of reproductive isolation before fertilization. So maybe you either block maybe mating to occur, just like the other two birds that they can't mate, can't interbreed because of song and behavior difference, or maybe there's some sort of blockage to fertilization such that the egg cannot be fertilized by the sperm. Or there's a second category called post-zygotic barriers, which maybe after fertilization, other things can happen that block this, the mature offspring from forming. So some hybrids, like we said, hybrid is the offspring from mating two different species. So some hybrids actually may have developmental errors, or they can also become infertile. Now, we already mentioned that infertile because we already mentioned that in order for it to become or is classified as two different species, they have to be able to, one same species, they have to be able to interbreed and produce fertile offspring. So if they're infertile, which means they cannot give birth to the next generation, then that is a barrier. Or have lower survival rate, like they will never make it to adulthood. That's also another source of barrier. So zygotic in these two words denotes the formation of a zygote. That's what it is. That's why it's called zygotic. And zygote is formed when sperm fertilizes an egg. We're talking about, the moment the sperm fertilizes the egg, we call that a zygote. Of course, it then will develop into other things. So a zygote will eventually develop into a mature embryo with time. So hence, prezygotic barriers are those that inhibit formation of a zygote. That's why it's called prezygotic. While postzygotic barriers are those that prevent the zygote from growing and maturing fully. Or even if they were to grow to maturity and are born successfully, they may not be able to survive or unable to produce offspring. So a couple things with the postzygotic barriers. So regardless whether they are prezygotic or postzygotic barriers, they both result in reproductive isolation, meaning that they cannot interbreed and they cannot produce fertile offspring. And hence formation of new species, that's how it comes about. This is what we call the process of speciation. Now let's discuss some of the prezygotic barriers that can exist first. So first, there can be habitat isolation. Two species that occupy different habitats within the same area is what we mean by habitat isolation. And on top of that, not only do they live in different habitats, they also rarely encounter each other at all, if at all. Okay? So that's what we mean by habitat isolation. For example, the apple maggot fly, actually right here, as you can see, actually feeds and mates only on apples and hawthorns as well. While blueberry maggot fly here mates and lays eggs only on blueberries. They don't mate or feed on any other fruits. Yet both of them live in the exact same geographic area. So if these two populations of species ever meet each other, they probably never will. They never will meet because they live in completely different habitat. That's what we mean by habitat isolation. They will not meet each other even though they live in the same area. They will never be able to produce interbreed and produce offspring. They will be unable to produce any viable offspring because of the fact that all its life, all this fly sees is apples and hawthorns. All this fly's life is sees blueberries. And they would never intermingle with each other because they don't leave their food source or where they mate. That's what we mean by habitat isolation. Second, there can be what's called temporal isolation. What do you mean by that? So it is when two species breed during different times of the day, or sometimes even different seasons, or different years, such that they cannot mix their gametes. So temporal goes with time. It means timing. If timing of reproductive maturation is not the same between two species, then they cannot interbreed with each other. So for example, the western spotted skunk mates in late summer, right here, while the eastern spotted skunk mates in late winter. both live in the same area, but again, they will never mate. Even if let's in this scenario, they live in the same habitat, they live in the same area, but they cannot interbreed because of the difference in mating season. So if it's off, there will never be mating between these two species. Third, there can be behavioral isolation. This is what it means is that even closely related species can have courtship rituals or other unique behaviors that are different. So that will, sort of tell the animal that you're a different species and we don't interbreed. Okay, so no interbreeding will occur when these small behavioral changes are different between two species. So, for example, a specific sound or call or even dancing style that is only unique to that species, they will not interbreed. Doesn't matter if they even look similar or live in the same habitat. Again, going back to those yellow birds we talked about earlier, the meadowlark. So it doesn't matter if they live in the same area or look the same. Okay. So for example, these are blue-footed boobies, and they actually mate only after a courtship display that calls for males to high step, which draws the female's attention to his bright blue feet, which I thought was pretty cute and cool idea. When you see this picture right here, they look like any bird that has these neutral coloration on their bodies, kind of like white and brown colors. And then all of a sudden, when you get to the bottom, their feet are this bright light blue color that looks like they're wearing blue socks or shoes or something. And if you're not looking at a cartoon picture here. This is a real picture of these blue-footed birds and boobies. So I thought I would show you their mating ritual, which was pretty interesting. This shows off their cute little blue shoes. There's that calling, and then there's that high stepping. High step, high step. Okay, again, these are from the Galapagos Islands. So let's see, before we get to the last type. The fourth type of pre-zygotic barrier is called mechanical isolation. It defines as two species. They can try to attempt to mate, but morphological differences, which means their shape, the way their body is structured, those differences prevent successful completion of the mating process. So one example is snails. So when snails, as you can see in these pictures here, when snails attempt to mate, they approach each other usually head first. When their heads have moved slightly past each other, so they here's a snail, here's a snail, just to be quick and simple. They move towards each other, and then they kind of move until their heads are past each other. And then the snail's genitals will emerge and only able to mate if their shells spiral in the same direction, because they will not come in any sort of interbreed or start the mating process, go towards each other in any other way other than head first. So that's why their shells had to spiral in the same direction if they wanted to try to mate. Now, if a snail try to mate with another snail where their shells spiral in the opposite direction, the two snails' genital openings will actually not align and mating cannot be completed. So this is how snails are able to keep the two species separate by this mechanical isolation, physical barriers that stop the species from interbreeding. The last type of pre-zygotic barrier discussed in this chapter is gametic isolation, which is, as the word suggests, gametic. Remember, gametes means sperm or egg. So it means that sperm of one species probably may not be able to fertilize the eggs of another species. So And there can be a couple reasons to this. First, maybe sperm cannot survive in the reproductive tract of the other species, or even sometimes, though in the wild, sperm is released into the surroundings, and supposedly the female will lay eggs, and then the sperm will get to the eggs. So maybe when the sperm is released in that particular environment, those sperm will not survive, or they cannot penetrate the egg membrane due to biochemical structures and differences. So, for example, sea urchins will reproduce by releasing sperm or eggs into sea urchins, release sperm or eggs into the surrounding water because that's where they fuse to form zygotes. So here's these beautiful red sea urchins and these beautiful purple sea urchins. And their gametes just can't fuse together because their surface protein bind very poorly to each other. So that goes with the inability to penetrate the egg's membrane. That's what keeps these two sea urchin species separate from each other. So even though they live in the same area, they release gametes in the same area at the same time. So it's not habitat isolation, it's not um, the temporal isolation. Zygote will still be unable to form in this scenario due to differences in their gametes that prevent fertilization. That's what we mean by gametic isolation. Now we move on to discuss some examples of post-zygotic barriers that prevent reproduction. So first, there can be reduced hybrid viability, which means that genes of different parent species may interact in ways that impair the hybrid's development or survival in either case. Both stops the hybrid from growing properly. Remember that hybrid means offspring from interbreeding of two species. That's what hybrid means. So for example, there are subspecies of salamander that we can see in this picture here. They live in the same area where they may occasionally mate. They do mate. They might have successful fertilization, successful offspring birth, but most hybrids do not complete development. And those that do are actually very frail. So this means that even though offspring hybrids can form with these subspecies of salamander, they either don't finish forming within the womb of the salamander, or for those that make it to live birth, they tend to be weak and die off. That's what being frail means. So that's why it's reduced hybrid viability. They won't survive due to those reasons we just talked about. Second, there can be what's called reduced hybrid fertility. This means that, let's say, if chromosomes of two parent species differ, let's say, in number or even structure, meiosis in hybrid may fail to reproduce normal gametes and therefore are infertile. Since no offspring can be produced from these hybrids, gene flow can't happen. And that's why we don't have separation. We can't have merging of the two species and keep them separate. So that's what we mean by reduced hybrid fertility. They become infertile. So for example, there are male donkey and female horse that will occasionally mate and produce a hybrid offspring called a mule. But mules, like right here, are sterile, which means they are infertile. They cannot produce sperm or egg. And same goes for hinny, which are actually hybrid from a female donkey and a male horse, and they produce also hinny that are infertile as well. So if hybrids are infertile, then they will never be able to give birth to the next generation of hybrids, which means there will never be a whole new population of hybrid species. That's how reduced hybrid fertility presents a post-zygotic barrier to having to keep it and keeping the two species apart from each other. Third, there can be what's called hybrid breakdown. Hybrid breakdown. So some first generation of hybrids maybe they are viable, they are fertile, but when they mate with each other or either parent species, we're talking about plants here, okay? They will mate with their parents for plants. Offspring are either frail or sterile. So, for example, strains of cultivated rice produce fertile hybrids in the first generation. So, this is the first generation. And then plants in the next generation, right here, this dinky little thing, are very small and sterile, which means they cannot further, you know, expand the population of the rice. So um, if the next generation again are infertile, then again goes back to the fact that they would never be a whole new population of hybrid species. Um, so that's what we call hybrid breakdown. Okay, these hybrids are able to mate with each other or mate with the parents in the plant's case. We'll talk about plant reproduction in the next few chapters. But so they are able to mate, but their offspring then are frail or sterile. In order for a species to become a completely separate species, population of species, they must produce viable offspring that is enough to grow a whole new population. Just a few is not enough. Just one generation is not enough. So remember that we have been discussing the biological species concept, where species are defined in terms of reproductive compatibility. New species are formed with the presence of reproductive isolation. So, hence, there are actually limitations to which the biological species concept can be applied because of that criteria. Well, first of all, reproductive isolation of fossils cannot be evaluated. So we can never, tell anything about species that is present in the fossils. We have to be able to see their behavior, observe their reproductive behavior in order to say that, okay, these are two separate species. Second, it does not this concept does not apply to organisms that reproduce asexually, because we've been talking about mating. That means that organism reproduce sexually. So if somehow these organisms reproduce asexually, for example, like prokaryotes or bacteria, then we cannot apply this biological species concept. Fossils can't be evaluated. Those that reproduce asexually cannot be evaluated. And third, for some species who are morphologically and ecologically distinct, they actually somehow do have gene flow, between them. Um, some exceptions. So, for example, there is the grizzly bear and polar bear, and they actually again will mate occasionally and produce hybrid offspring called the broiler bear, which have been seen in the wild and able to produce the second generation. So, if you look online or go to the textbook, you will be able to see this picture of the hybrid broiler bear, um, which is pretty interesting. But they are morphologically distinct, ecologically distinct, but somehow they do mate and produce an hybrid. So again, this will be then where the biological species concept cannot be applied, because these two, even though they produce hybrids, these two bears remain as distinct species. So there are other speciation concepts that are useful for specific situations. Kind of like most things when we talk about abstract biological theories and concepts. So if this doesn't work this way, well, maybe there's other explanation to these exceptions. So here are two of those examples of species concepts that can explain those things that biological species concept can't talk about or evaluate. First, there is the morphological species concept, which actually distinguishes a species by body shape and other structural features. Now, it can be applied to both asexual and sexual organisms, and useful even without information on gene flow. The disadvantage, though, for this particular concept is that it kind of relies on subjective criteria. So it's morphological species concept again distinguishes or separates two animals or organisms as two separate species just by looking at their body shapes and structural features. So but this is kind of where we can see how it can be subjective, because whether one says, oh, this part of the animal looks similar or different from another animal, that can be subjective depending who's looking at it. Somebody may say, oh, that looks different. Another person may actually say, oh, they look the same. So that's the downfall of this morphological species concept, but it does apply to both asexual and sexual organisms. Next is the ecological species concept. So this is where it defines species by its ecological niche, how members of that species interact with non-living and also living parts of their environment. That's what we mean by ecological niche. So for example, there are two species of oak trees that are different in sizes or their ability to tolerate dry conditions will actually, and because of that, those differences, even though they're different, they will occasionally interbreed. Even though they're different, they will interbreed. But because they occupy different ecological niches, they are actually considered as separate species, even though they're connected by some gene flow. So in this case scenario, you have two separate species of oak trees, but they also tolerate things differently, but somehow they do occasionally interbreed. Um, but they occupy completely different biological or ecological niches, and that's why scientists say, "Oh, okay, they are different species because of that." So this concept also applies to sexual and asexual organisms, and it emphasizes the role of disruptive natural selection as organisms adapt to their different environments. So it can be applied to both, and then emphasizes the role of disruptive. What do we mean? What do we mean by that? Well, we have talked about what is disruptive selection back from the last chapter, where the environment will favor both extreme phenotypes over the intermediate ones. So in this case, there are more than, there are a lot of oak tree species, and it is because of their differences in ecological niches. You may have one that favors one extreme at the same time, also favoring the other extreme of phenotypes. Okay. All right, we have talked about this. There are more than 20 actually other proposed species definition, and the usefulness of each definition of these species concepts will depend on the situation and the research questions being asked. So there are many other different ones that we won't have time to discuss them all. So that's why we just brought up two other different ones that are applicable to things that applicable to things that biological species concepts can't. But for this chapter, we'll mainly focus on the biological species concept. Just bringing up some few ones for you to see what other ones are like. There are two other speciation processes we will discuss in this chapter. First of all, those are called allopatric speciation, and the other is called sympatric speciation. This is where unique species can arise from existing species by processes that may involve geographic isolation, hence allopatric speciation, and those that may not have geographic isolation, which is sympatric speciation. In both cases, we know that gene flow is somehow interrupted. So now you have two separate species. But it's just how they become two separate species is what these two different processes are differing in. We will talk about these two selections now in detail with these two speciation processes. First, we'll start with allopatric speciation. Allopatric literally means other country, which means that you have two different countries. So allopatric speciation is when new species arise due to a population being divided into geographically isolated locations because they are completely separate from each other and there's no way to overcome or outrun this obstacle or barrier. Their gene flow is interrupted, and that's how you can have development of two separate species. For example, in this particular picture right here, if a lake has a barrier like this one right here, that it was at first underwater, and then let's say somehow there was a natural event that led to a decreased water level where this water level will drop. So, and that would actually lead to two or more smaller lakes form. So now we're not looking at sympatric yet, we're looking at allopatric right here. So if water levels were to drop down to here, let's say, at the top of the barrier, now you have this part of the environment and this part of the environment being geographically isolated or separated from each other. So now two smaller lakes are formed and populations are now geographically isolated, separate from each other. So another situation where allopatric speciation can arise is when parent individuals colonize some remote area away from the central area, let's say, and descendants will be geographically isolated from the original population. For example, the flightless cormorant. We said that there was this cormorant that once flew, and then they flew from South America onto the Galapagos Islands, and then eventually, with time and many, 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 many generations after, they eventually lost their wings because that was not critical for survival in this particular Galapagos Island environment. And that's how you have this, again, parent individual, the original cormorant species, colonizing some remote area, which is the Galapagos Islands, and they isolated them geographically from the original population, which is those that were from South America. So even though they're coexisting species, they look very different from each other due to this allopatric speciation, okay, geographically isolation. So the next question to ask is, how formidable must a geographic barrier be to promote allopatric speciation? Does it have to be? I mean, is there quantification to it? Oh, yeah, they allow like only 1%, 2% of the animals through, so it's a good allopatric speciation, or does it have to be 100% blockage? Well, it turns out that it depends on ability of organisms to move about. Can this organism cross a river? So if there a river all of a sudden starts to form between two separate geographic areas, then that's a geographic isolation if they can't swim. Can these animals climb over mountains? Let's say a long time ago, all of a sudden, a small little mound of mountains start to form, eventually leading to a hill that they can't cross. This particular animal can't cross the other side. That's also a geographic barrier. Can seeds be blown away? Seeds are blown away in a certain way, and that is a type of gene flow. But let's say their seeds are very heavy, or that they're embedded in the ground or something, so they can't be blown away, then there is no gene flow between species. So in order for two species to form, in a sense, there needs to be some sort of blockage to gene flow between the animals or organisms. So, whether a geographic barrier is enough to produce allopatric speciation, it all depends on how the organisms are able to move about or able to cross that particular barrier. As long as there is a barrier where two population species cannot cross over to the other side, then there can be allopatric speciation. So for allopatric speciation, once geographic isolation has occurred or has formed, separated gene pools may start to diverge, because now they live in completely different areas, and mutations can happen, natural selection can happen. When we're talking about that, flying to a remote area, natural selection can definitely favor other things that are different from the original population. Um, and all this genetic drift can also alter allele frequency, as we've talked about, which is completely random with genetic drift, whether there's a sudden random catastrophic event that has caused these alleles to change. That's how eventually these two populations will diverge, and that's how we form these separate species. Now, these things, whether mutation, natural selection, or genetic drift, they were mechanisms that we discussed in the last chapter. So here's an example of allopatric speciation. These are called mosquito fish, which actually colonized series of ponds that later became isolated from each other. There's no gene flow that can occur between the ponds. There's no way they can escape or go through a tunnel or something and mate with each other. So there is no interbreeding that can happen between the ponds. And the difference is that between some of the ponds is that some ponds contain predatory fishes, while some ponds don't have predatory fishes. So ponds with predatory fish, sort of favors a mosquito fish with body shape that enable very rapid bursts of speed, while ponds without those predatory fishes, they actually favor body shape. They improves the ability to swim for long periods of time. So here's that mosquito fish with predators, so their body shape kind of goes with that burst of speed. That's why they're larger. Their muscles are more massive because that's how you're able to produce those sudden burst of energy. If taken anatomy before, we've talked about marathon runner versus sprinters. In this case, we need sprinters to sprint really quickly away from predators. That's why they look larger. Whereas those without predators, their body shapes should sort of favor this long, steady swimming, which is going with the analogy of marathon runners. They're there for the long haul, swimming for long periods of time, and that's why their bodies tend to be more slender. They're not looking for that burst of energy that we need. So these fishes that live in the ponds with predators were able to accelerate their escape a lot faster than the fishes that live in ponds without predators. And hence, those fishes that live in ponds with predators also had a much better survival rate when placed with predators. So here are the two comparisons where we have the source of mosquito fish that was with predators present, and here's the one without predators. We're talking about how they're able to accelerate, how fast they're able to accelerate. And you can see that the source where mosquito fish were living with the predators present, they're able to escape a lot faster, more than 50% than those without predators. Survival rate, how about with predators? That's also dramatically increased in the mosquito fish that had predators present because of their ability to have that burst of energy to escape. Whereas those without predators, not really high survival rates with presence of predators when they dump them into water body that have those particular predators. So thus, different body shapes have evolved in mosquito fish populations, simultaneously by selecting for fishes that can accelerate to escape predators. So that is what the selecting factor there is, that there is either predator or no predator. And that's why their body is different by natural selection. In this case, it is due to geographic isolation. That's why we call it allopatric speciation. Now, on the same note, has this also affect evolution of reproductive barriers? Researchers have actually found that female mosquito fish also prefer to mate with males with body shape that is similar to their own, which means that speedy females only prefer to mate with speedy females. So even though we call this allopatric speciation from geographic isolation, this also created a reproductive barrier because of the differences in their ability to speed, where mosquito fish that live in ponds with predators now will not mate with those that live in ponds without predators because of their biological differences that has evolved over time with these two different geographic areas. On the other hand, sympatric speciation, sympatric means same country. This is when new species arise even though they live in the same geographic area. So how can this happen when organisms are living in the same area? Because when we're talking about geographic isolation, that makes sense because they are living in two separate areas. So what can cause gene flow to stop when two species are actually living in the same area? Well, gene flow can actually be reduced by other factors other than geographic isolation. There can be a couple things: polyploidy, sexual selection, and habitat differentiation. We'll talk about each of these in detail right now. So let's talk about polyploidy first. What do we mean by that? This is when organisms contain extra sets of chromosomes. Hence, poly means many. Ploidy is, when we talk about haploid, diploids, um, refers to how many sets of chromosome it has. So polyploidy means many extra sets of chromosomes, two or three or more in this case, due to accidents during cell division that can happen. So it turns out that the polyploidy is more common in plants. 80% or more of plants alive today are actually formed by polyploid speciation. We're not talking about humans. Turns out that polyploidy in humans can lead to early miscarriage, usually within the first trimester, 7 to 9 weeks, and they actually will not develop into an embryonic maturity for it to have been born, because when humans have more than two sets of chromosomes, we're talking about more than 46 chromosomes, that's a lot. And they actually it will be so chaotic with the DNA sequences that the embryo just won't survive. So when we're talking about polyploidy as a way of speciation, this is more commonly referred to in plants. So there are two types of polyploidy in plants. One is called autopolyploid, which means it is an individual with more than two sets of chromosomes. Um, Two sets of chromosomes derived from a single species. That's why autopolyploid. Auto usually means on its own, it is able to produce more chromosomes by itself. That's what auto usually means, automatically on its own. So for example, from a diploid to a tetraploid. So tetraploid is, we're talking about four sets of chromosomes, can produce fertile offspring by either self-pollinating or mating with other tetraploids. Because we talked about how in order for us to classify two different or populations as separate species, is that they have to be able to produce viable fertile offspring. So it turns out that even though you increase the chromosome sets from diploid 2 sets to 4 sets, they can actually sort of self-pollinate and mate with other tetraploids to produce more population of themselves. And that's why they can have a new species arise from having, even though they have more than 2 sets of chromosomes. But tetraploids are actually reproductively isolated from the diploid. Because even though you're like, well, diploid had became a tetraploid, maybe these tetraploids can mate with each other, but can they go back and mate with the diploid to produce more species? They cannot. Okay, so they are reproductive isolated from the diploid, which actually limit the amount of gene flow that can happen between these two species, and that's due to their offspring usually have reduced fertility. So tetraploids can mate with tetraploids, but if tetraploids try to mate with the diploids, their offspring can't be fertile. So that's why they cannot form another new species. So regardless, whether without any, where we're talking about without any geographic isolations, autopolyploidy can generate new species in just one generation. How does that happen? Here is a picture that describes that. So here you start with a diploid cell, which has 2 sets of chromosome and a total of 6 chromosomes. Because of, let's say, some cell division error, they have produced a tetraploid cell that now went from diploid to tetraploid. So these tetraploid species can then generate gametes that are diploids, because usually with diploid cell, they produce gametes that are haploids. So in this case, they are able to now produce gametes that are diploids. These diploids can fuse and mate with each other to produce cells with new species that now have tetraploids. Okay, so these are gametes produced by tetraploids, and you now have a whole population of species that all have cells with tetraploids or four sets of chromosomes, and they do produce these viable offspring and cultivate its entire population, a whole new population of tetraploids. Second type of polyploidy is called allopolyploid. Allopolyploid is when an individual with more than two sets of chromosomes are derived from two different species interbreeding, which actually produce hybrid offspring. Now, these hybrid offspring again usually is infertile at first, but again, just like autopolyploids, they may actually be able to propagate itself asexually by self-pollination. So, for example, a diploid have six chromosomes breeds with another diploid that has four chromosomes. So the diploid with six chromosomes will produce haploid gametes with three chromosomes, while the diploid with four chromosomes will produce haploid gametes with two chromosomes. So I know that sounds like a whole mouthful. So here's a diploid cell. Okay, it is a diploid with two N number and has total number of six chromosomes. When this cell produce gametes, remember they produce haploid gametes, so they produce gametes with three chromosomes, half of that. This diploid cell usually have a 2n number of four chromosomes, so when they produce gametes, they will produce haploids that only have two chromosomes. This gamete have three chromosomes, this gamete have two chromosomes, is what we're talking about in this situation here. So when these haploid gametes fuse together or fertilize each other, it will generate a hybrid zygote with five chromosomes. So this is three, this is two, added together, they have this new organism that is made up of new organism that has five chromosomes. Now, usually this zygote will be sterile because their chromosomes are not homologous, meaning that they are coming from two separate gametes that are different from each other, so they cannot pair up in this particular hybrid to form a viable individual. And because they're sterile, they're not homologous, they cannot pair during meiosis. So this particular hybrid zygote may actually be able to reproduce asexually on their own if there was what we call if there was a mitotic or meiotic error. Okay, so a mitotic or meiotic error must be present in order for this sterile hybrid to now be able to reproduce on its own asexually. So this hybrid will somehow, all of a sudden, on its own, double that chromosome number from 5 chromosomes to 10 chromosomes. And now we can have generation of a new species where the diploid number is 10 chromosomes. And this is what we call an allopolyploid. It is originally, this hybrid species is from one species that has 6 chromosomes, another species that has 4 chromosomes. They fuse together. Supposedly they are sterile because they can't form homologous pairs. But all of a sudden it just decides to, by some mitotic or meiotic error, decides to duplicate each one of these 5 chromosomes. Now you have homologous chromosome to pair up with, and now this has become a viable, fertile diploid cell offspring. Okay, that hybrid that can go on to produce its own progeny of 10 chromosome offsprings. That's why it's called allopolyploid. So in the wild, 3 species of a plant called goat's beard with a genus name Trigopodon actually live in the same area. And they were able to produce 2 new species by allopolyploid speciation. So here is the 3 species that live together, and this one area was called T. dubius, T. patensis, and also T. periphrolius. Okay, periphrolius. T representing Trigopodon. Okay, so T. dubius and T parenthesis and T peripheralius were three different species of these Trigopodon that live in the same area. T dubius and T pratensis both were actually yellow flowers, as we can see here and here. And they both actually have 12 chromosomes. But when they went through the allopolyploid process, they were able to produce a viable hybrid offspring species called the T macellus. And they actually contain 24 chromosomes, going back to that same story about the whole process of allopolyploid formation. And that's how we get this new species of T macellus that has 24 chromosomes. This new species of T macellus is able to grow its population by either asexual reproduction or by the continued hybridization between T dubius and T pratensis. All in all, we have a whole brand new species, sympatric speciation from allopolyploidy. Okay? And scientists later also discovered that T dubius, which have this bright yellow color flowers, and T perifolius, that have purple flowers, also formed a new hybrid species called T mirus. T mirus right here. And they produce pink flowers and also have 24 chromosomes. Well, it turns out that many, important agricultural crops, other than these goat's beard wildflowers producing all these two new species, either T macellus or T mirus, both completely different amount of chromosomes from their parent species. One even looks different from their parent species. They found out that this actually happened quite often in plants. The agricultural crops such as oats, cotton, potatoes, tobacco, and weeds are all polyploids. Okay? Today, the plant geneticists actually can create new polyploids in a lab because if it wasn't created in a lab, they have to wait for all these events to happen in the wild, and mitotic and meiotic error may not happen very quickly. So they actually are now able to create these new polyploids in the lab by using chemicals that will induce these meiotic and mitotic errors instead of just waiting by chance for them to happen. So in a sense, we have taken the original evolutionary process as seen in nature that we just talked about with allopolyploid, and now we use that same method to produce new polyploid species where we can now insert our own wanted beneficial traits, such as large size of fruits and seeds. Maybe there's this a good crop out there that is very valuable and very helpful to the whole population of humans for consumption. But yet this particular fruits or plant is very small, or they're hard to cultivate, so they're very expensive. Now, if we can mass produce these fruits or plants and crop somehow in a large number, and also they're bigger in size, we can feed a lot more people at lower cost. That's how we can use lab techniques to induce polyploids that have these particular traits that we want. There's also the ability to produce polyploids that are able to form this resistance to specific diseases. So let's say there's this disease that constantly kill off this particular crop. So the yield is not high for this agricultural crop. So if we can induce or have this resistance to diseases gene into this particular plant by polyploid mechanism, then this plant can now be able to survive and have a much higher yield, again lowering the overall ultimate cost of the crop, and it's not so expensive anymore. Some can actually even have tolerance to specific environmental conditions. They say they will ripe faster or able to tolerate cold temperature, because usually with crops, that particular plant can only grow best under specific environments, temperature, soil differences, air differences, etc. So they can actually create polyploids that can tolerate specific environment conditions, and many, many, many more traits that we can introduce into plants for benefits of the whole agricultural business and also the human race. So it is about an hour right now. I'll stop here and continue to part two of the video.